For my local expert project, I'm going to tell you all about stem cell culture. Stem cell therapy is something that really interests me, so I was quite pleased that it tied into our cell culture module and I could do a deep dive on it. Before we talk about culturing stem cells, you have to know what stem cells are. These are undifferentiated cells that have the potential to develop into many other types of cells within the body. There are two different kinds of stem cells: human embryonic stem cells, or HESCs, also called pluripotent stem cells, which have the ability to differentiate into all the cells of the adult body. Somatic stem cells, which are the stem cells that adults have, are also called non-embryonic stem cells. These are found within the body in a tissue or an organ and can differentiate into the specialized cell types of that tissue. However, in 2006, scientists managed to devise a way to take some adult human cells that are fully mature and reprogram them back into a stem cell that resembles an embryonic stem cell and can therefore differentiate into any cell type in the body. These are called induced pluripotent stem cells, or iPSCs. So, how do we grow stem cells in the lab? Just like culturing any cell, we need culture medium or nutrient broth. So stem cells are incredibly fickle, and the slightest variation in culture media changes them. This means that the media needs to be optimized for growing different types of stem cells, and this media should be changed each day. Stem cells are grown on plastic culture dishes coated with extracellular matrix components such as gelatin, matrigel, or collagen, or with a feeder layer to aid in attachment. HESCs are traditionally cultured with mouse embryonic fibroblasts, or MEFs, as a feeder layer. In addition to aiding in attachment, feeder layers also prevent uncontrolled proliferation by utilization of factors such as leukemia inhibitory factor, which is a cytokine released by MEFs. However, this can also cause issues, as HESCs cultured in the presence of animal products express the non-human sialic acid NuGC. In addition to a feeder layer, a culturing media needs to be provided, which is often fetal bovine serum, or FBS, but it can also be an artificial replacement, such as synthetic serum substitute, knockout serum replacement, or StemPro. The simplest culture medium developed by scientists is called E8 medium and contains only 8 essential elements: DMEM F12 medium, selenium, sodium bicarbonate, L-ascorbic acid, transferrin, insulin, TGF-beta1, and FGF2. This medium doesn't do anything to make them differentiate, so they can just hang out and proliferate really well without having to move the cells forward into differentiation, which makes things more efficient. The creators proved this with over 25 passages over more than 3 months with 2 HESC and 5 iPSC lines. During culture, stem cells need to be replated as the cells divide and get overcrowded, usually repeatedly over many months. Each cycle of subculturing is referred to as a passage. At any stage in the process, batches of cells can be frozen and shipped to other laboratories for further culture and experimentation. So what happens once we've grown enough stem cells? Well, like I said, stem cells are incredibly fickle, and the slightest variation in culture media will change them. This makes initiating differentiation in some forms pretty easy, but it also leads to a lot of inconsistencies. So again, you've taken differentiated, fully mature adult cells that have been reprogrammed back into the pluripotent state to create iPSCs. This was achieved over several weeks by forced expression of genes that are known to be master regulators of this pluripotency. When the iPSCs have grown in culture under the appropriate conditions, as discussed on the last slide, they can remain undifferentiated. Then, to generate cultures of specific types of differentiated cells, you can do one of 3 things: change the chemical composition of the culture medium, alter the surface of the culture dish, or modify the cells by forcing the expression of specific genes. Directed differentiation is designed to mimic signals that are received by cells when they undergo successive stages of development. The extracellular microenvironment plays a significant role in controlling cell behavior, so by manipulating the culture conditions, it is possible to restrict specific differentiation pathways and generate cultures that are enriched in certain precursors. Most directed differentiation protocols are formed to mimic the development of an inner cell mass during gastrulation. During this process, pluripotent stem cells differentiate into ectodermal, mesodermal, or endodermal progenitors. There are numerous protocols about the methods of forming progenitors of cells of each of the germ layers, such as cardiomyocytes, hepatocytes, renal cells, lung cells, motor neurons, intestinal cells, and chondrocytes. Unfortunately, there is no shortage of limitations to stem cell culturing. HESC differentiation must be specified to avoid teratoma formation. If the cells are not given the proper conditions to differentiate, you're going to end up with cancer. Ethical considerations need to be taken into account, especially when working with human embryonic stem cells. In talking specifically about utilization for therapeutic treatment, the patient is usually not getting cells that originally came from their own body and were induced back to stem cells, but instead are coming from a different person, and therefore rejection is possible. Many things add up to make this expensive. The inconsistency of differentiation due to environment specifics, it being hard to mimic the natural microenvironment, it's very labor intensive, it's hard to optimize, and it also yields heterogeneous populations only. Obviously, there aren't only downsides to utilizing stem cells, or they would be virtually obsolete in the lab. These cells are very useful for understanding the basic biology of the human body. Embryonic bodies, which are spontaneously differentiated human embryonic stem cells, can be studied instead of embryos or animals to predict the effects of a treatment on early human development. They are utilized for drug toxicity testing, helping move new therapeutics to market more quickly. Current possible stem cell therapies and treatments for spinal cord injury, heart failure, retinal and macular degeneration, tendon ruptures, and diabetes type 1. There have also been great outcomes in utilizing stem cells on patients with osteonecrosis of the femoral hip as well, which we'll talk about in a minute. When it comes to transplants, there are many limitations of traditional transplantation, the number one of which is the lack of available organs to transplant. For example, with stem cells, transplantations to assist with hematopoietic diseases such as leukemia is a matter of growing new heme cells instead of harvesting them. However, transplantation therapy, for the most part, is going to be tied to two other similar techniques to stem cell culture: 3D culture and organoid culture. These techniques rely on stem cells at their base, but they take the science further. While I have no doubt that one day within my lifetime we'll be able to grow entire new organs for transplantation from stem cells, right now such experiments are still in the preclinical phase, while numerous stem cell therapies have already occurred. There are many quality control tests run to make sure that the stem cells are not contaminated and have not been differentiated. The four you see here on the screen are just a very small but important sampling. As previously mentioned when discussing restorative medicine treatments that stem cells have been utilized in, one such treatment is for hip osteonecrosis. Traditionally, cord decompression, or CD, is the treatment method, but in Sen et al. 2012, they showed that the utilization of a stem cell treatment in addition to CD had far better clinical outcomes. This figure here shows magnetic resonance imaging of an osteonecrotic hip in a 32-year-old man, both before operation and 1 year post-op. You can see the reduction in the volume of osteonecrotic area after the combined treatment. iPSCs were utilized to create cardiomyocytes for patient-specific cardiotoxicity drug testing, drug discovery, disease modeling, and regenerative medicine in Burrage 2013. The author created a cost-effective protocol for an in vitro cardiac differentiation that is effective for a wide variety of stem cell lines. This protocol produces contracting human embryoid bodies, or HEBs, with near total efficiency in less than 9 days, and minimizes the variability in cardiac differentiation commonly observed between various stem cell lines. In this figure, we see in the very top row the media utilized and the timing of the media changes. In the middle, directly under the timeline, the physical task performed at each time point can be seen. Underneath this, and above the microscope images, is the cell format showing the move from T25 flasks to V96 plates and U96 tissue culture treated plates. The microscope images then show the growth of cells from original pluripotent cells at 100% confluence all the way through day 9 of growth and the clear differentiation that is being performed. Kai et al. 2007 reported a novel method to efficiently direct the differentiation of HESCs into hepatic cells in serum-free medium. This protocol utilizes activin A, fibroblast growth factor 4, and bone morphogenic protein 2, among several other medium components, to mature the HESCs into liver cells expressing the appropriate markers and functions. Figure 1 shows the 3-stage differentiation strategy on the left, as well as the immunofluorescent results of the mature liver cells expressing the appropriate markers to the right. Thanks for learning about stem cells with me today. I really hope you enjoyed it.